to continue with our discussions with the culture and the medium. So, we say culture is a term given to microorganisms that are cultivated in the lab for the purpose of identifying and studying them. So, because there are microorganisms that are fastidious or need special nutrients, and non-fastidious which does not need special nutrients or media. Okay? Medium is the term given to the combination of ingredients that will support the growth and cultivations of the microorganisms by providing essential nutrients required for the growth. Okay? Kasi ang different organisms require man sang different nutrients in order to grow. Okay? So, multiplication in order to cultivate these microorganisms in large number to study them. So, take note, every microorganisms, there are different nutrients or media being used in order them to grow. So, microbial culture, which are used for the growing microorganisms such as bacteria or yeast. Okay? The most common growth media na ginagamit class is the nutrient broth and then the agar plate. So, nutrient broth, ito yung solution form. Okay? Specialized media are sometimes required for microorganisms and cell culture growth. So, depende yung specialized, depende yan sa microorganisms, okay? Or sa specific microorganisms based on their growth also. Next, culture media used to grow bacteria can be used to enrich the numbers of bacteria or to increase the number of bacteria, select the certain bacteria and suppress others. Kasi take note, kasi yung selective muna media, example, specific na sa Pseudomonas, so yung hindi Pseudomonas, hindi siya makasurvive doon sa media na yun. Okay? To prevent contaminations and to come up with a successful growth and number or large number of the bacteria of that specific bacteria to be studied. Kailan na isuppress mo yung others, ha? Hindi pwede yung Pseudomonas plus Staphylococcus. So, iba-iba yung culture media kasi magkaroon ng tawag natin na cross-contamination dyan. Okay? So, pwede mamatay yun si Pseudomonas, pwede mamatay si Staphylococcus aureus. So, differentiate among different kinds of bacteria. Okay? Need for culture media. It is usually essential to obtain culture by growing organism in artificial medium. That's why we use agar. Okay? Yung nutrient agar is very common. If more than one species or type of organisms are present, each requires to be carefully separated and isolated in a pure culture. That's why pure culture, kung damo na, example, Staphylococcus, Strep, or Escherichia coli, so they have to be separated and isolated in different culture or media. Okay? So, basic requirements for culture media, we have energy, carbon, nitrogen. So, mineral salts, we have sulfates, phosphates, chlorides, carbonates, potassium, magnesium sulfate, magnesium carbonate, calcium carbonate, okay? So, suitable pH is 7.2 to 7.4. Take note. It is almost weak base or weak base or weak acid sa water is weak base. Okay? So, growth, so almost neutral man, 7.2, di ba? Growth factors, we have arginine for E. coli, glutathione for gonococci, we have cholesterol for mycoplasma, aryl sulfate amide, atypical bacteria, glycerol, mycoplasma hominis, sulfonamides, rickettsia rickettsii, tryptophan, salmonella typhi, we have the L-cysteine for listeria monocytogenes, sodium chloride for Vibrio parahemolyticus, and factor X and factor 5 for Haemophilus influenzae. So these are needed for the growth of this specific bacteria. Ang tawag ng growth factors. Next, classification based on physical state. We have liquid media, semi-solid media, and solid media. So based on the presence of oxygen and reducing substance with aerobic and anaerobic. Mga baka-aerobic, may oxygen. Yung anaerobic, walang oxygen. Based on nutritional factors, simple, complex, synthetic, and special media. Okay? So, ano yung mga special media? We have enriched media, enrichment media, selective, differential, indicator, transport, and sugar. So for simple media, we have simple basal media are composed of the minimum adequate nutrition for the growth of the bacteria. Okay? Example, nutrient broth or agar. So mayroon siyang peptone water. Example niya ng mga tawag ng simple media. Composition, so pwede yung Lemco, peptone, sodium chloride distilled water. Okay, 10 grams ng Lemco, 10 grams ng peptone, 5 grams ng sodium chloride, and 1,000 ml of water. So when 2 to 3% agar is added, then we have it nutrient agar. For semi-solid, the agar concentration is 0.2 to 0.4. Uses, ito yung basis ng most of the media used for study of common pathogenic bacteria. Yung mga common Staphylococcus aureus, ganun. Okay? It is used for subculture of certain organisms. Okay, ito yung nutrient broth and we have the nutrient agar. Ito yung pinaka-common. Ginagamit natin doon sa mga stuff. Okay. So, peptone, we have the basic liquid media, the clear colorless watery usually in test tube yan. So, ano yung content niya? Peptone, sodium chloride, and water na 1 liter. So, uses of peptone water. So, the media is used to chiefly as a basis of carbohydrate fermentation process or fermentation media. So, nutrient broths may be contain small amount of sugar derived from meat. And yung mga meat, yung protein, pwede mo yung convert into sugar, ha? It is essential that the basal medium, various carbohydrates are added for fermentation test should be free of natural sugars. Okay? Ano yung mga different natural sugars that are present, okay? Example, yung mannose, natural sugars yan. Okay? So, free from natural sugars. Kasi kung may mga natural sugars, may tendency magkaroon ng natawag natin na it will hinder, the fermentation process. It is also used to test formation of indole, culture of organisms for demonstration of motility. So, kung say motility, may mga movement. So, pwede mo yun magitawag natin ang peptone water. So, makita mo yan to examine under microscope. So, gamitan na yan ng biconcave na slides. So, lagay natin ng culture doon ng peptone. For example, kung ano doon, may mga motility or movement, of the bacteria. Okay? Complex media have added ingredients for bringing the certain properties of bringing out properties or providing special nutrients for growth of bacterium in question. So, you have synthetic media. So, these are prepared from pure chemicals, exact compositions of the medium very well known. For example, yung Dovus medium. Okay? So, later on, we'll discuss some composition of this one. Semi-defined media, ito yung media that exact chemical composition and is not known because substance like meat and peptone use. Diba? So, yun ang tawag na semi-defined media. So, most of the culture media used for routine diagnostic work are semi-defined culture media because mostly kung meat and peptone, ito yung needed ng this is needed by the bacteria for growth, especially protein. Okay? Special medium, we have enriched media. When basal medium is added with some nutrients such as blood serum or egg called enriched media, they are used to grow bacteria which are exacting in their nutritional needs. So, yung tawag na ito, enriched media. Example yan, Dorset's egg medium. So, base structure creamy, slope kept screw cap bottle ito. Selected medium for isolation, usually inagamit ito for Mycobacterium tuberculosis. So, ito yung hen's egg and nutrient broth. Yan ang composition ng inatawag natin na Dorset's egg medium. So, blood agar, ito yung enriched media. So, usually, blood agar is red. Composition, sterile nutrient agar plus deprobenated sheep blood. Okay? So, routine culture. So, widely used medical bacteriology. Okay, usually sa mga medtech. Showing hemolytic properties such as Streptococcus pyogenes, beta hemolytic siya o alpha hemolytic. Okay? So, blood agar ang ginagamit dyan. We have chocolate agar, also called heated blood agar. So, enriched media, chocolate brown color. Okay? So, ang procedure niyan, melt the desired amount of nutrient agar, cool in a water bath, 75 degrees Celsius, then 10 ml of sterile blood, allow the medium to remain in 75 degrees Celsius, percent, sorry. So, usually makita mo parang luto blood dinuguan, pag i-heat mo na ang blood, usually ang itsura na do chocolate na. Pero kung tamaan po i-heat, maging black na yung blood. Amo man na sinugaluto dinuguan na babalaan mo na amo na siya ang kumbaga amo nang itsura niya. Kung tamaan siya kaluto ang blood, ma-black siya. Pero kung tama-tama lang siya, pagbutang mo, chocolate ang color niyan, chocolate brown. Okay? So mixing the blood and agar by gentle agitation from time to time until the blood becomes chocolate brown in color within about 10 minutes. Then pour into the plates. So usually ito ginagamit sa Neisseria, Haemophilus, and Pneumococcus species of bacteria. Okay? So ano yung enrichment media? In this media, it has a stimulating effect on bacterial to be grown in or inhibits its competitor or other bacteria. This result in absolute increase in number of wanted bacteria related to other bacteria. Example, ang ano mo lang ang gusto mo isolate is Salmonella typhi. So you have to use the enrichment media. So example niyan is selenite F broth. It is enrichment medium culture of Salmonella typhi and Paratyphi bacilli from stool sample. So pwede mo siya i-count yung sample mo sa stool, then use this one. Then hopefully 90% or 90% sina Salmonella typhi ang ma-isolate or Paratyphi. Okay? Principle, so neutral pH, solution acid salinity has high toxicity to coliform group of bacteria and not most of Salmonella. So usually kasi kung sa tae manggod sa stool sample, damo may expected na may E. coli siya. So toxic yung salinity ng acid mo o salinity ng imo na media, toxic sa mga E. coli. But ano naman siya, advantage ano naman siya, prepared naman na ano yan na environment yan sa mga Salmonella group. That is why yan ang ina-isolate ng Salmonella kung suspect ka na may Salmonella typhi ang patient. So usually stool sample ang kuhaan mo using the selenite F broth. Okay? So this one selenite alkaline peptone. Okay? So we have selective media. So this one is the medium which certain substances are present, okay? Inhibit all other bacteria except for desired bacteria. It encourages growth particular species from mixed inoculum.